Hello friends, hope you all are doing well. This is Dipsika and today we will discuss syllabus of MSc Zoology semester 1. In semester 1 we have 4 papers, paper 101, 102, 103 and 104. 104 will be practical. First of all we will see pattern. There will be two examinations, mid-semester examination and end-semester examination. In mid-semester examination, we have two groups of questions of 20 marks. In group A, there will be 5 questions. It may be a multiple choice question or fill in the blanks of 1 marks, and group B will contain descriptive type 5 questions of 5 marks. Out of 5, we have to write only 3, and end semester examination will consist of two groups, group A and group B. Group A will contain two questions. Question number 1 will be of very short type of 5 questions, and each question will consist of 1 marks. Question 2 will be of short answer type and it will be of 5 marks. Group B will contain descriptive type questions. There will be 6 questions of 50 marks, and out of that we have to only attempt 4 questions. In paper 1 we have systematics, evolution and bioinformatics. In systematics, animal systematics, we have to study basic concept and nature of taxonomy and systematics, contribution of systematic to biology, different types of classifications, numerical, phonetic, cladistic, evolutionary systematics, concept of cytotaxonomy, chemical and molecular taxonomy, systemic hierarchy, names, codes, operative principles of nomenclature, applications of important rules. And evolution, unit 2, evolution, concept of evolution, theories of organic evolution, new Darwinism. Synthetic theory of evolution. These two topics, concept of evolution, theories of organic evolution, we have already discussed. You can go and check out in channel. Population, gene frequency, Harding-Weinberg's law in genetic stability, genome evolution, evolution of multi-gene family, genetic drift isolation. One by one, we will discuss all these topics and finish paper 101 of semester 1. And bioinformatics, third unit is bioinformatics, principle of bioinformatics and its application, biological databases. In the biological databases, we have to study 4 databases: nucleic acid sequence databases, protein sequence databases, protein structure databases, literature database. Data retrieval system, search engine, entrance, molecular sequence analysis software package and tools. In this, we have to study BLAST, RASMOD, and Biologist Workbench PERL. And in paper 202, we have to study invertebrate diversity and quantitative biology. In invertebrate diversity and quantitative biology, we have 2 units. Means in paper 102, we have 2 units that consist of invertebrate diversity and quantitative biology. In invertebrate diversity, we have to study concept of protostomes and deuterostomes, origin of coelom, acoelom, pseudocoelom, schizoscoelom, enterocoelom, locomotion of protozoa, locomotion in Nidaria, Annelida, and Echinoderms with reference to hydrostatic movement, origin of segmentation, expression and osmolation in protozoa, nephridian coelomic system in annelids, excretion in arthropods, respiration, arthropods, molluscs, concept of host specificity and host parasite relationship. In Unit 2, we have quantitative biology, means biostatics. In biostatics, we have to study samples and population, sampling designs, probability distribution and their properties, and normal probability, binomial probability, Poisson distribution, all these 3 types of distribution we have to study. Hypothesis testing. There are 2 types of hypothesis testing: non-parametric test and parametric test. Chi-square test, GTF test, analysis of variance, correlation and regression. We have to study all these things. Evaluation of biodiversity indexes, Sanon-Vano index, index of dominance, similarity and dissimilarity index, association index, 2 into 2 contingency table. And in paper 103, we have biotechnics, histology, and histochemistry. Means in paper 103, we have two units biotechnics and histology and histochemistry. In biotechnics, we have to study spectrophotometry and spectrophotometric principles, spectroscopy, atomic absorptions, EASR, NMR spectroscopy, microscopy and scanning and transmission electron microscopes, fluorescence microscope, crystotechnics, cryopreservation of cells, tissues, and organisms, cryotechnics for microscopy, separation techniques. We have different types of separation techniques: paper chromatography, TLC, GLC, ion exchange, and HPLC. We have to study electrophoresis, centrifugation, basic principles, differential and density gradient centrifugation, immunocytochemistry, and ELISA. In histology and histochemistry, we have to study fixation and tissue processing, types of fixatives, chemistry of fixation and selection of fixatives, dehydration, clearing and amending, microtomy, staining of paraffin sections, principle and method of staining, histological stains, histochemical identification and localization of the following glycogen, glycoprotein, protein, and end groups. Means we have to identify and localize mercury, bromophenol blue, linhydrin ceph, performic acid ceph, and performic acid alcine blue, lipid mutase, Benzedum black B method, Sudan 3 and Sudan 4, Nile blue sulfate method, nucleic acids, DNA and RNA by methyl green, proline Y, DNA by fusion reaction. In and paper 4, we have practical, and you can see the pattern of practical in this and practical तो बस एंड टर्म में होगा and this will consist of 100 marks. You can see in pattern passing mark is of 45 marks, and this all questions you will have to attempt this 1 2 3 4 5, and record and sectional work is of 10 marks and viva was 20 marks. And you can see the syllabus, and you can prepare your practical file according to syllabus. Okay, thank you guys. Hope you are clear with semester 1 syllabus. Thank you. Take care. Bye bye.